Thank you, Jolie. I'd like now to introduce Amber Kuliev. He's the Director of Research at the Reproductive Genetics Institute in Chicago, and he's a clinical professor at Florida International University. Amber Kuliev received his PhD in clinical cytogenetics in '69, and from 1979 he's been the head of the World Health Organization Hereditary Disease Program in Geneva, which initiated the introduction of CBS for prenatal diagnosis, which started the development of genetic risk approach to common disorders in the early '80s. Dr. Kuliev, together with Dr. Berlinski, were the founders of Reproductive Genetics Institute in Chicago, which introduced preconception genetic diagnosis in the early 19th, and one of the pioneers to use polar bodies for chromosome analysis and gene defects. RGI, Reproductive Genetics Institute, also initiated the establishment of human embryonic stem cells with genetic and chromosome disorders, which currently constitute the world's first and largest repository of embryonic stem cell lines with normal and abnormal genotypes. Dr. Kuliev was also the founding scientist secretary of Preimplantation Genetic Diagnosis International Society, and is presently the executive director of this society, as well as the secretary of ISRM Special Interest Group in PGD. He has published more than 200 papers, and today he will give us, with his perspective, the history and evolution of the technique.
Thank you, Sandy, for the introduction. It's, of course, a privilege for me to talk about evolution in my capacity from the very beginning at running international working group on preimplantation genetics, which was established in 1990 at the first PGD Congress in Chicago. We were running this predecessor organization of Preimplantation Genetic Diagnosis International Society with Yuri Verlinsky, whom unfortunately we lost 4 years ago. I started this first slide showing this technique that was born 20 years ago. Which one is it? This one. It doesn't go. Can you change? Okay. So from this slide, you could see that from 1990, when PGD was introduced by two groups in London, by Handyside and by Yuri in Chicago. So you could see that until the birth of this technology, we had only few cases have been done. And apparently only few births, and we had probably not more than 10 cases altogether. And after the introduction of this technique, and it was very clear the need for this technique, because the IVF was extremely inefficient, and in order to improve the efficiency and standard of IVF, we needed some tool in order to select the embryos which have more potential to go into pregnancy. For this reason, of course, the birth of these technologies was very important. And if you see even the first, even within the first year, from 134 cases that we summarized, actually it is the fourth international meeting of preimplantation genetic group that was hosted by Jamie Griffo, Santiago Mone and Jacques Cohn in 1994, which was summarized already. As you could see, we had already 25 cases after the start. And at that point, we had only one birth, but quite a few ongoing pregnancies, 5. And the distribution of them was, as you could see, that the majority was cleavage state, have done by Cornell group, and we have done polar body cases. And this distribution shows that these two groups, we started introducing this technique very actively. So if you look at the membership of the International Working Group at that time, you could recognize many leaders in this field now, starting from Urey, Handyside, Griefer, Monet, Cohn, and Mark Hughes, and Julie Simpson. So this was a group that actually documented the first results of this technique, which was published in Journal of Assisted Reproductive of Genetics in 1994. The next step was, of course, we have to mention in 1995. It was the introduction of this technique to chromosomal rearrangement. It was Santi's prize paper in SRM, which he reported based on the fact that the first polar bodies consisted of metaphase chromosomes. So he was very right just to go into that and to, based on the fact that if you find in the first polar body both derivative or both normals, you will see on the left side, you would select the oocyte with the normal distribution. So on the left side, you will see it. On the upper, in those cases that you could find the one derivative and one normal, of course, in this case was unbalanced. But what Santi at that time could recognize, but we recognized it later on, and Santi himself was found that in those cases that you have two different chromatins, it's difficult to do. And this was because, in the contrary of the classical understanding that bivalence going to one side or another, it appears that each chromatin goes independently to different sides. So for this reason, in the middle, you could see that the polar body has the two chromosomes, both of them with the different chromatids. So in this case, of course, you cannot identify on the base of the first polar body unless you go to the second one. And you could see, in case you have the normals or the balance, you know that it's normal. So this is one of the first cases that we have done. And it's very interesting that what we found that in the translocation between chromosome 15 and 1, you could see on the top right, that both of these consist of two chromatids. One chromatid have two signals, red and in the red arrow, red and blue, when the other one also has the two chromatids different. So in this case, of course, on the right top, you could see that in the second polar body, you could see the normal distribution, so that we confirmed this on the cleavage stage. On the bottom, you could see that this shows that probably the second polar body was containing the balanced, so that we have the normal chromosomes in the cleavage stage. The next step was, of course, introduction, very important stage when the 5-color probes was introduced. The importance of this was because it's commercial. So it started with very active introduction in the IVF clinics. And of course, this is one of the cases, just as an example, you could see on the polar body that on the top left, you could see that only on the arrow shows only one signal for chromosome 21, which inferring that you have the Down syndrome in the embryo, because the second polar body has the normal distribution. So based on that, of course, we started the active introduction application in the IVF. And in a few years, we have opportunity to get together 3 leading expert laboratories. And we summarize the first, it's published in Fertility Sterility, the 2 groups, 3 groups, including the Cornell group. At that time, it was all the San Barnabas, Chicago, and at that time already Luca Generali came to the field, as he has done more than any center in Europe was doing chromosomal testing. So the result showed that the first over 1,000 recognized pregnancies was summarized in this case. So we have actually documented the first thousands of babies born at that time. And of course, the next 10 years, there was the efforts to test all the 24 chromosomes. There was attempt to do it by phase rehybridization in 1 and 2 and 3. There was also attempt to do all 24, which was not practical, of course. And at that time, when I was demonstrating here ERASE-CGH, which we have done first in the next step, which is not in the graph, of course, but this is the 2009 as the first experiment we have done with Bluenome, in which we validated that if you have this first polar body normal on the top, second polar body on the middle, and you have, in this case, of course, the normal oocytes. And the most important from this was that this technology was capable of detecting not only chromosomal non-disjunction, which is only 90% of origin of chromosomal disorders, but also chromatidal malsegregations, which is showed here, the chromosome 16. So this was a very important point, which showing that it is quite important technique, and this was introduced very, very actively. And according to the Bluenome, you could find out that now some centers doing between 500 and 1,000 cases a year. So it's, if we are talking about the 1994, when we have only 25 cases, now we probably have over 100,000 of cases has been done with probably dozens of thousands of children born. And of course, the graph also not contains the introduction this year of next generation sequencing, which was publication, the deliberation was reported this summer that the first baby was born after NGS used for chromosomal testing. And of course, again, this publication or deliberation was Doug and Wells and Santiago Monin, collaborative work. And I would like now to conclude with this slide. And of course, Santi mentioned, and I'm sure that in this audience we are going to hear more and more about the screening. But from the, on behalf of the International PGD International Society, I could refer you to the guidelines that the screening was never used, because screening in classical genetics means that you identify the risk group and you require obligatorily the definitive diagnosis on the risk group. Otherwise, this is not screening, but it is diagnosis. And there is no any excuse to make mistake, because any mistake from that, you are going to be liable for this mistake. That's why it is not, of course, screening, it is diagnostic, because on the basis of this test, you transfer the embryo, you are liable for your diagnosis. And this is the most recent ICRM glossary, in which you could clearly see that the PGD used for genetic abnormalities as well as chromosomal abnormalities. The same in the Pre-Implantation Genetic International Society. We use the terms PGD for chromosomal aneuploidy or pre-implantation aneuploidy testing. Thank you.